Okay. That leads us to the next thing about regulation of regulation by non-covalent binding. And that's where one protein binds and regulates another. Once again, when one protein binds another, the surface area of contact between the two proteins is often pretty significant. So many times that can induce dramatic structural changes in the protein that's being regulated. Often more so than what you can get by just sticking a small molecule or an ion somewhere on the protein. We have numerous examples. Many different proteins are regulated by many different other proteins. A couple examples, once again, just to reiterate what we already had before. Active G proteins. They work by binding non-covalently and regulating a whole slew of target proteins. Since there are many different kinds of G proteins around, that means many different kinds of target proteins can be regulated by G proteins. That's one example. Another example is calmodulin. When you have calcium ions bound to it, 4 of them in particular, calmodulin works when it's active by binding and regulating a whole bunch of other kinds of proteins. Now, once again, when we go through these things, a given protein can be regulated in multiple different ways. The same protein might be regulated by other proteins, might be regulated by binding of small molecules, and or might be regulated by covalent modification. So many proteins are regulated in multiple ways by multiple regulators. And we'll have a tie-in example towards the end of this class. Okay. All right, questions on that? That kind of covers our regulation by small molecules, or rather by non-covalent binding. The next thing we're going to look at is regulation by covalent modification. In other words, we're going to attach something to the protein with covalent bonds. And covalent bonds are strong. They don't shake off. They don't break easily. So, covalent modification. Okay, a couple things about it. First, some generalities. We're going to attach a regulatory molecule by covalent bonds. Unlike non-covalent binding, covalent bonds are strong. They don't break very easily. Protein's not likely to shake something off that's attached by a covalent bond. Now, that leads to a couple of things here. First thing, covalent bonds don't just happen on their own. To attach a regulator to a protein with covalent bonds, that's going to require a condensation reaction. What that means is attaching something by covalent bonds is an endergonic reaction. So, when you attach something covalently, you have to do a condensation reaction. What that means is two things. It takes energy to do this. it's not going to happen on itself. It's going to take an input of energy, usually ATP hydrolysis. Not always. And the second thing is this. That's going to be a coupled reaction. Your energy source, whatever it is, whether it's ATP, hydrolysis, or whatever, your energy source and your substrate, your target protein, you're going to have to do that at the active site of an enzyme. It's going to take a special enzyme to perform this covalent modification. So it requires an enzyme. So you have to have an enzyme to do that. Now that once it's done, it's stable. It's not going to break on itself. But the problem is when you want to reverse it, when you reverse the reaction, it's not going to fall off on itself. It's on its own. It's going to be attached by a covalent bond. So to do the reverse reaction, that's a hydrolysis reaction. Now, hydrolysis reactions are exergonic, but the problem is it requires a second set of enzymes to catalyze them. So in effect, every time you do or reverse this, you require a dedicated set of enzymes to perform either the condensation or the hydrolysis reaction. Now, downside of this is it takes time. You have to call up those enzymes to do this thing, and then however long it takes the enzyme to actually do the reaction. So it's going to take time. A slow response is not something you do in a millisecond. It's going to take seconds to minutes to do that. And likewise, it's going to take an equal amount of time to reverse it. So covalent modification is best used when you want to make a stable change to a protein. So covalent modification is best for long-term regulation, where you actually need to do a stable change here. Oh, I'm sorry.
Okay.
Okay. All right. Well, there's several major kinds of covalent modifications that occur to proteins, but we're just going to mention the two most important ones. And we'll look at one in particular, a little more detail, to explain the pluses and minuses and sort of the details about it. What the details about this also apply to the second one we're going to look at. But the major one is what we call phosphorylation. And phosphorylation is the covalent addition of phosphate ions to a protein. Now, phosphate ions have that powerful double negative charge. So you can imagine if you stick a phosphate ion on a certain part of a protein, that's going to have a real significant impact. Negatively charged amino acids will be repelled from it. Positively charged amino acids will be pulled towards it. Hydrophobic amino acids will see that and just run, duck, and cover. They want to get out of there. They want to get away from that. So phosphorylation is the covalent addition or removal of phosphate ions. The second one we're going to look at is acetylation. Acetylation is where you covalently attach what's known as an acetyl group. An acetyl group is basically acetic acid covalently attached to something. You got two carbons, organic acid at this end, and sometimes you lose that hydrogen and picks up a negative charge. So acetyl groups tend to have a negative charge again. Once again, that's going to play around with a protein structure. So those are the two major, there are a couple others, but those are the two major types of covalent modification. And we'll take a look first at phosphorylation in a little more detail. Because phosphorylation is one of the major means of protein regulation. So we'll take a look at that in a little more detail in just a moment. Hopefully you're recording this and not